I think we are a little bit more than 2 minutes into the hour, and I think we are almost ready to get started. Santi, you ready? Great. All right. Well, welcome everybody to join us today at the July GemPharmatech webinar. My name is Jing Li. I'm the Director of Life Science and Technology at GemPharmatech US. Today is my great pleasure to have my colleague Santi Chen to share her insights on immuno-oncology and oncology in general. Before we start today's session, I'll give a brief intro of Santi. Santi is a research scientist and business development professional with over 10 years of experience in autoimmune diseases, immunology, and oncology. Santi received her training in human B-cell development at the Govan Institute of Medical Research, Australia, in immunology, followed by a postdoctoral fellowship at the Children's Cancer Institute, Australia, focusing on pediatric leukemia and therapies. Her projects included efficacy evaluation and mechanistic investigation of a variety of testing agents. Her research projects resulted in multiple publications in journals such as Blood and the Journal of Immunology. He has also received rewards national rewards and international awards. Santi joined GemPharmatech not long ago. She is our Associate Director in Immunology and Oncology. So today, welcome Santi to share her insights with us. The topic for today's discussion is challenges and strategies of preclinical evaluation of NK cell-related immuno-oncology therapies. So without further ado, Santi, take the floor away.
Thank you, Jing, for that very kind introduction. I will stop my video just to save the bandwidth. Okay, so let me get myself situated here. Again, thank you, Jing, for that very kind and generous introduction. And thank you, everybody, for attending this webinar. I'm very excited to share some insight about preclinical agent evaluation of NK cell-related immuno-oncology therapies. And the intent of this webinar is to make it as a discussion and a food for thought on what is the best models or design for preclinical agent evaluation. So let's get started. So NK cell is the rising star among cancer immunotherapies. This schematic here shows you the growth that is expected from this NK market. We can expect an exponential growth in the next years, a few years, and with the forecast of compound annual growth rate of approximately 17%. So NK cell immunotherapies, there are different approaches, and I will describe a few here, and I wouldn't be surprised if in the coming years that there will be more approaches for NK therapies. So the first one is utilizing their antibody-dependent cell cytotoxicity. So typically in cancer cells are very smart. They can exhibit inhibitory receptor that if it is engages with cells such as NK, it will inhibit their cytotoxic function. So there are strategies, for example, antibodies that would inhibit the inhibitory receptor, allowing the activation of NK cell, and then ultimately the cancer cell will die. Another way is using bispecific or trispecific, and we'll describe this a little bit further later in the talk, where the bispecific will identify, for example, the tumor antigen as well as the NK, bringing the two cells together so the NK can perform its cytotoxicity function. Another way that's been employed by various pharmaceutical companies is genetically engineering NK cell. And then infusing this in patient or in preclinical setting as well that's undergoing evaluation. A newer kind is using cytokine such as IL-15, which is a very important cytokine for NK function, and I'll describe this again further in the talk. One of the one that's excited about is about superagonist IL-15, and I'm very excited to see the development of superagonist IL-15 in the coming years. So to understand about NK cell, let's take a step back and look at the key features of NK cell. So in this schematic, it shows the NK cell development. At the top is the mouse development, and down the bottom is the human development. In human, it can be identified via CD3 negative, CD56 positive. It makes approximately 5 to 15% of human peripheral blood and 2 to 5% of murine splenocytes. It is involved in cytotoxicity against injured cell or virus-infected cell or cancer cell without the need of prior sensitization. And I think this is one of the key features of natural killer cell. You usually express surface marker CD16 or Fc gamma receptor 3 and CD56 in human, while in mice it's NK 1.1 or 1.2. It doesn't express T cell receptor or B cell receptor, and it carries its function in both the innate and the adaptive immune regulation. So as I mentioned earlier, NK cell are key effectors of anti-tumor response using a variety of methods, whether it be direct cytotoxicity, ADCC. It also recruits other members of the immune cell, for example, T cell, DC, which eventually leads to the activation of the adaptive immunity via different cytokines such as TNF-alpha, IL-8, and so on. It also releases the exosome. So some examples of NK cell related therapies. First is drugs that target inhibitory receptor on NK cell. As I mentioned earlier, tumor cells are very smart. They can express inhibitory surface marker that will stop NK cell from carrying its function. So agents such as leronlimab here, which is a fully human IgG4 antibody, inhibit this interaction, allowing NK cell to be activated through the KER blockade. Another example here, very similar design, NK here on the left and tumor cell on the right. Here we can see the anti-NKG2A antibody is inhibiting this recognition, allowing the activation of NK cell. Another example of NK cell related therapies is NK cell engager. So this is for the ADCC function. There's bispecific and trispecific. I want to highlight with trispecific, typically there's IL-15 that is also engineered together within this tri-antibody, which supports the NK cell survival and expansion, as well as the ADCC function. This is another example here from Sanofi, that's currently in phase 1/2 clinical trial. If your agent requires CD16 receptors, I highly recommend you to look at our previous webinar. We have a humanized Fc gamma receptor mouse that would be excellent for the agent evaluation that requires CD16 recognition. And if you scan on this QR code here on the bottom right-hand corner, it'll bring you to the webinar link. Another example is using direct NK infusion, whether it be in autologous fashion, allogeneic fashion, or engineered NK cell fashion. So at GemPharmatech, our goal is to create mouse model that will support the evaluation of various NK cell related therapies, and also to understand about the NK development. So we do this by two ways. One is genetically engineered mouse model, or what we call it as GEMS, and the other one is NCG-hIL15. I will briefly touch upon the first point here, the genetically engineered mouse models. We have over 20 humanized mouse model that can be used to study NK research. So here on the schematic, NK cell, we can see very complicated a variety of receptors, cytokine, and so on. So we have multiple humanized mice, as you can see, it's listed in here, on variety of mouse background. So I invite you, if you're interested in any of this, please feel free to scan this QR code, have a look at our website, and also contact us for more information. So for the purpose of this talk, I will delve deeper in the NCG human IL-15. So one of the challenges in preclinical evaluation of NK cell therapy is the limited persistent in vivo. As I mentioned before, IL-15 is critical for the development and persistence of NK cell. Meanwhile, mouse NK poorly supports human NK because of its low affinity to the human IL-15 receptor. So there are strategies that's been employed to overcome this limited persistence. One is using exogenous cytokines, so multiple cytokine injection. Alternatively is engineering NK cell so that it can produce endogenous cytokine to support itself, or making the pushing the NK to be more mature. So a couple of examples here. So the first one, as I mentioned earlier, is multiple NK cell infusion. So this study was published in 2021, and this particular investigator utilizes multiple injection of NK cell. Another way is multiple NK cell infusion plus exogenous cytokine injection, such as IL-2 or IL-15, such that employed in this by this particular investigator. One of the issue of the exogenous cytokine injection is the risk of adverse reaction. So it is important that the cytokine concentration is not too high, but it's also not too low, so that it can support the NK development and function. So because of that, we designed an immunodeficient line, which is the NCG line that expresses human IL-15. So we denote this as NCG human IL-15 here. You'll see this over and over again in the subsequent slides. In the initial validation, we look at the mRNA expression across multiple organs, and we compared it between the NCG in black and the NCG-hIL15. As you can see, it's quite comparable across the board. Most importantly, looking at the protein expression, here we can see there is no human IL-15 in the NCG, but in the heterozygous and the homozygous IL-15, we can see the human IL-15 expression. So this would be a great model to support a variety of NK reconstitution study. So this can be used for pure NK or CAR NK evaluation. Studies that require PBMC transfer, for example, if you're evaluating antibodies that targets NK or T, and also CD34 HSC reconstitution, again, for antibodies that target T, NK, or T. So you'll see this summary table in the subsequent slide as well. So I will describe, to begin with, is the CD34 HSC reconstitution. So we found using this model, the NCG-hIL15 exhibit extended lifespan after CD34 HSC reconstitution. So on the right here, on the left here, we can see in black is the NCG CD30 reconstituted with CD34 HSC, and in red is the IL-15 NCG reconstituted with CD34 HSC. You can see that the body weight increases, the mice very healthy, and a very long survival curve. You can see this is closer to 150 days. This would be a great model that requires long study window. We also found that this model promotes NK cell development. So again, in black is the NCG reconstituted with CD34 HSC. In red is the IL-15 NCG reconstituted with CD34 HSC. Very clearly, you can see that there's faster engraftment in the IL-15 model and also higher engraftment. Looking at the T cell, we can see very comparable percentages here, also similarly with the CD4 and the CD8 cytotoxic cell. For the B cell, we can see a decrease, and this is expected behavior. This is not the model for B cell reconstitution. However, we are developing more and more NCG line that can support the B cell. Most importantly, for the purposes of this webinar, we're talking about the NK, is looking at the CD56, which is the NK marker. And you can see here in the NCG, it does not support NK, but in the IL-15 mouse, we can see an increase, so approximately 20% NK cell. So one of the important thing is, while this shows great number, but we want to make sure that this NK cell are functional. So we took it one step further and we look at the inhibitory and activating markers. So the KIR3DL and the NKG2D. So at the top here, this is the NCG parental reconstituted with CD34-HSC. And down the bottom is the IL-15 reconstituted with CD34-HSC. You can see very nice NK numbers here in comparison to less than 1% in the NCG. And you can see that there's the functional markers still present. We took this an extra step further by taking the NK cell that was reconstituted in this mouse model, take it out of the splenocytes, purify it, and then we did in vitro ADCC assay. In this case, we combine it with the K562 cancer cell line in 2:1, 5:1, and 10:1. You can still see cytotoxicity. And we also take it with Raji cancer cell line and tested it in the presence of rituximab. And again, you can still see the cytotoxicity. Looking at it in vivo, it was tested with Raji in this case, and it was tested with rituximab and Blincyto. And you can see a very nice tumor growth inhibition. So the NCG-hIL15 reconstituted with CD34-HSC are ideal models for anti-tumor efficacy evaluation of antibodies that target, that modulate the NK function. So I've described to you about the human HSC reconstitution study in NCG-hIL15. So let's now go back and look at the human NK and human PBMC. So we did a comparison where we reconstitute the NCG-hIL15 with human PBMC and compare it with purified NK from PBMC. And here we can see that in this model, the mice is very healthy. There's a little bit of GVHD, but it's only score 1. Long survival rate, so this was tested across 40 days, and increasing body weight. Looking deeper into the percentages at the top here, and down the bottom is the counts per cell, per mil. And here again in black is the PBMC, so it's a mixture of population, and in red is the pure NK. And you can see that there's, even though the percent of CD45 is lower in the pure NK, but you can have a look here in the CD56 positive section, it's almost 100%, which is expected because this is a pure NK cell. Again, highlighting that the NCG-hIL15 is a great model for NK study. This is also reflected in the count, cell count per mil. So you can see there's a higher percentage in the pure NK. The PBMC still supports it as well. A little bit lower because it is a mixture of population, but we notice that this tends to increase over time. One of the key points that I want to highlight in human PBMC reconstituted model is donor selection and screening. So in this figure here, we tested 4 different PBMC donors. And what we notice is different donors behave very differently. So I'm going to highlight this figure here in the middle, looking at the CD56. You can see that different donors have very different NK reconstitution levels. So the black one is middle. We see higher in the donor number 3 and 4, and much lower in donor 5. If your agent is critical with the percentage of NK cell, we highly recommend that you do donor screening. and select the appropriate model. We've also noticed that different donor can induce GVHD faster and others much slower. So depending on what your need is, if you're using human PBMC, we highly recommend that you do screening. So I want to highlight this interesting publication that was published in 2021 that shows the application of our NCG-hIL15. For this particular investigator, they took Mia-Pakas, a pancreatic cell line, and then injected in vivo, and they gave multiple CAR-NK injection. And here they were comparing the NCG in blue to the NCG-hIL15 mice. And here you can see that the CAR-NK number is much higher in the IL-15, again, supporting the fact that it supports, this is a great model that supports NK persistent. And with the increase of cell CAR-NK number, we also see greater tumor growth inhibition. And again, this is also reflected in the increased cell survival rate compared to the NCG itself. One of the interesting information from this published paper is that it supports long-term human NK persistence. So one of the things they did is they took purified NK from PBMC, it was engrafted in NCG line, and they look across 12 weeks, so a time course study, and they compare it with the NCG-hIL15. In the NCG alone, because of the lack of IL-15, we expect to see the IL-15 and the NK cell drops over time, and this is the case. But in the IL-15, we can see that there is increase of NK cell, again, highlighting the fact that this model supports NK development, but you'll see that it drops over a period of time. However, you can see within the first approximately 10 weeks, it remains at about 10 to 15%. So this is still a great percentage for NK study, also a great study window. Our speculation, this drop is because the IL-15, at some point when the NK continuously proliferate, it takes up all of the IL-15 around and depletes that resource, and that's why we see the decrease here. However, overall, we can see that the IL-15 remains healthy, so an increase in body mass and long survival. This is also to highlight that in the IL-15, it shows great tumor growth inhibition. So I hope I've convinced you that the IL-15 is a great tool for preclinical agent evaluation that requires NK presence. You can use this in a multiple different fashion, whether it be for CAR-NK evaluation or mouse reconstituted with human PBMC or CD34-HSC. So the take-home messages for this, the NK cell is a rising star in cancer immunotherapy. There's multiple pharmaceutical companies are modulating NK function in a variety of different ways. For example, the anti-NKG2A that inhibits, that blocks the inhibitory receptor. IL-15 is extremely important for NK cell development and function. And the NCG-hIL15 is a good model for preclinical agent evaluation that requires NK cell presence and function. So I just want to quickly touch upon one extra point. As I mentioned earlier, at GemPharmatech, our goal is to continuously create better mouse model that will allow humans immune cell recapitulation in vivo. So this schematic is our suggestion on which NCG is best used for various immune cell research. So for today's talk, I mentioned about the NK, so the NCG-hIL15. But as you can see, we have others. So we have the NCG-X that is useful for CD34 reconstitution without irradiation. We have a cross between these two, so NCG-X-hIL15. We're also working on a few other NCG line that supports B cell. We have a myeloid model, so NCG-SGM3, and a variety of models that supports the T cell. as well as quite a few that's a little bit outside of this area, but we're working on multiple aspects. So with that, I'll end my talk, and we're very excited that we can be a partner of your studies, and hopefully we can accelerate your innovation. Thank you very much, and I'll pass it all back on to Jean.
Hey, Santi. Well, thanks for the discussion and then the elaboration on your experiences and the insights on this topic, putting in the current status of the field. If I can ask you to bring back your own images as well, or yourself, let's open the discussion. So I do have a question in the Q&A box. So for the audience, if you have questions, feel free to drop it there. I'm gonna bring the first couple of questions that are already there to Santi, and I do see one of our audience is raising his or her hand. I will ask you to open your mic if our IT guru, Jeremy, behind the scene can enable that. So after the two questions, I'll come to that. So the first question to Santi, is human IL-15 evaluated in plasma?
I believe in the initial validation it was, and that was one of the data that I showed earlier. That was evaluated, and yes, these slides will be made available. I think Jeremy, our IT, is working behind the scene to make it available after this webinar.
Okay, let me check on the hand. I think the hand is down, so I assume you answered your question. So that's great. Let me check one more time. There's one more. Yeah, we answered that question on the slides. So I'm seeing another one came in asking about, it says, you showed the L2 and the L15 in one of your slides that both can support NK cell. Can you discuss a little bit more on how the L2 and the L15 work in this area supporting NK, and how is their function in terms of supporting CAR NK, if there's any information you can share?
Yeah, IL-15 has been gaining a lot of interest, and yes, it's IL-15 is not the only one. IL-2 can also support NK cell, and we are working on NCG-hIL15 that is in the works. We're doing a lot of validation. IL-2 has been employed by various studies as an exogenous cytokine, as I showed you in some of the slides. It definitely is an interplay. I think the key here is trying to find that balance between the IL-2 concentration and IL-15 concentration, so that it's not too high that it causes a lot of adverse event, but it's not too low that it won't show any effect. So we're definitely working on that, and hopefully we'll have it in the coming months.
Okay. I think I'm going to ask the follow-up question on behalf of the audience. Do you know how many months that is, or how soon that can be, come to the to the customer-facing perspective?
Yeah, we have some preliminary data right now. I emphasize on the preliminary here. I think one of the issue that we have with our IL-2 model is it's an overexpression system. So it appears to be too overexpressed, that it's causing a lot of adverse event. So we're employing a different strategy where instead of overexpression system, it's a knock-in system. And we're hoping that would alleviate that problem.
I see, great. So add on to that discussion on the L2, I want to share with the audience, although Santi didn't share the strain strategy of the NCG human IL-15, that model is a knock-in into the mouse locus. So I believe to some extent that strategy helped and ensured the expression level is physiologically relevant in the studies that we have been discussing. I saw a hand just now, but it went away. Oh, I see the question here. How many human NK cells in blood or IL-15 levels are physiologically relevant?
Yeah, in terms of in the blood, we know that in normal human, it ranges between 5 to 15%. In terms of IL-15, it can range, particularly if you're healthy or if you're a cancer patient. We know if you're a cancer patient with some cancers, it can go 50 above picogram per mil. So our model is, it's also around about that range, so it's physiological relevance. But I know that there are certain cancer where IL-15 is completely depleted, and that can be used as a biomarker. So I think the application of it is will be different depending on the type of tumors.
Great. Another question I'm seeing is talking about GVHD. Can you elaborate a little bit more on the CAR-NK therapy and how, in that context, the GVHD, whether that's a concern or not a concern?
For the GVHD, definitely if you're working with human PBMC, that is always a concern. That's why I highlight the donor screening is very critical. We don't see it so much with NK, but you know, even though you use a kit and purify the NK, you can never get 100% purity. And depending on how long the study goes for, that risk will not be 0%. So it's always something good to keep in mind.
Great, thanks. One more question. Have you ever thought of working with cord blood NK cells?
Yeah, for the CD34 HSC reconstitution, that's cord blood donor. If it's to purify the NK out of those, I think it's doable. I think the challenge there is getting enough cell numbers, because the cord blood typically is already very low volume, very limited volume, which is not like PBMC. Plus you need to purify the NK out of that. So the challenge there is potentially purifying the NK from the cord blood and then expanding it in vitro using a variety of methods before re-infusing it in vivo.
Got you. I think this is the last one I'm seeing in the chat, in the Q&A box, which is talking about there are CAR-NK therapies that target CD22, CD19, BCMA, and so on. Do you have corresponding models?
Yeah, that's going into the B cell section, BCMA, the CD19. We're working on a model that can support the B cell. So that's not an easy one. I think with B cell, there is so much interplay of receptors, cytokine stages, that it may take a little bit longer, but I know that our team is working very heavily trying to improve the B cell. So stay tuned. Hopefully in the coming months, we can do another webinar specifically on B cells.
Great. So I don't see any more questions in the Q&A box. If that's the case, I'm gonna say thank you, Santi, for the discussion presentation, and thank you everybody for joining us today, as well as your questions. Always good to chat with you and hear your feedbacks and opinions. So with that, I'm gonna say thank you, and we'll see you next month in August. Bye.